Hi everyone, and welcome to a BioColor webinar, Is it safe to dye? My name is Riikka Alanko, and I am the interaction coordinator of the project. Today we have an interesting set of speakers and presentations focusing on the theme of the safety of the dyes and dyeing industry. So we are covering, for example, the question what kind of harmful effects dyes can have on the environment, ecosystems, and human health, and how research and regulations can lead to more safer solutions in dyes and dyeing. Here you can see the detailed program of our webinar. So the webinar will be 2 hours long. We have 4 presentations, and we also have time after each presentation for audience questions. So you can leave your questions and comments through the chat section in Zoom, and I'll be monitoring those during the presentations, and we'll have a chat with the presenter here after his or her presentation. So let's keep the chat active, and of course there might be experts on the lines, so you are very welcome and free to chat with each other in the chat section as well. So for the first presenter and presentation, we'll have Riikka Räisänen, the leader of the BioColor project, a researcher at University of Helsinki, and she will describe more about the aims of the BioColor project concerning toxicity and safety of the dyes. So welcome, Riikka.
Okay, thank you. So hello everybody, and you are very welcome to this event, and great to hear and see that there are actually people participating all over the world. So it's really great that we will have something interesting to say for people from different continents. Okay, we have this BioColor project that we got started last year, and our aim is to seek solution to textile and material coloration. And as we know, dyes are used everywhere. We have been asking consumers what do they know or about the dyes or colors, dyes behind the colors, and many don't even think about where the color actually comes from. Anyhow, the global market value of dyes and pigments is really huge, and it's still growing all the time. And increased demand is especially in textiles, but also in paints and coatings and construction. So every day, huge amounts of dyes and pigments are used in different applications. The price of raw material at the moment is mostly the oil-based smaller compounds like benzene and so on, and this determines the price of the dyes. But also as the biodegradable material production increases, there has been search for different solutions for the current dye markets. So mostly currently reactive dyes are used for textiles, and their market value is a little bit bigger than for inorganic pigments because they are more expensive to produce. But when we think about the volume, then the inorganic pigments are the largest group of colorants used. And globally, the production of dyes is in hands of big companies. So there are hardly no more than 10 big companies. So it's very concentrated field of production. And of course, then there are some smaller companies, but most of the dyes it's concentrated, and a lot of dyes are produced in Asia. What do we know about the production and properties of dyes? What need to be known of dyes in order to develop something new and different? So starting points for organics, if we think of organic dyes, are simple aromatic compounds such as benzene, for example, and in those synthesis of dyes, very often catalyzed are needed, and also heavy metals such as zinc or chrome are used as catalysts. And in producing them, water, acids, and alkali are used as solvents. When we think about the process, then we must keep in mind that in organic system, usually the yield is not 100%. So every time we get less end product than what we would think about in optimal conditions. Of course, then since there are international agreements about, for example, these hazardous chemicals, and for example, this manufactured registered substance list, more and more purification is done for synthetic dyes. So the new synthetic dyes are more pure than those the previously one, which is of course a good thing, because very often those impurities might be hazardous. What we need from dyes and their properties is that they need to be stable. So the more stable the dye is, the more the better it is resist, for example, the use phase, and for example, the light and UV radiation, and the color in textiles or in surfaces will remain the same. Also, the dye needs to have affinity to the substrate, good exhaustion, so that a lot of dye goes into the fiber instead that it stays in the dye liquor. And of course, when we think about the use, so that the fastness properties are also good in washing, for example, for textiles. And when producing a dye, a safety profile is important, which describes the safety is aspects related to that compound, and Andreas will soon tell more about that. So dyes are stable, and dyes, but on the other hand, dyes and their degradation products may cause hazards to humans and environment, and this is one of these, or these are the aims why Biocolor project wants to develop colorants from biological sources. So mainly we think about biodegradable materials, but we also want to develop methods for cleaner and sustainable production processes. In production, dyes use biopathways, for example, in plants and microbes. Also, fungal engineering could be used. Efficiency in utilizing side streams and other industries in obtaining biomass from where the biocolorants could be extracted. We want to use waterless or low water processes, and of course study the fundamental understanding, to study the properties fundamentally of these dyes and processes. And finally, we probably have safe dyes and pigments to be used. And therefore, we know that when something is natural, it doesn't mean the same as non-toxic or safe, and therefore we need these studies of toxicology. And today we will hear more about the topic by the presenters. Okay, thank you. And I give the floor to Andreas.
Thank you so much, Riikka. So to proceed with our program, next I will welcome Andreas Uphoff, Scientific Officer at the European Chemical Agency. And he will talk a bit more about legislation and safety issues related to bio-based colorants. So welcome, Andreas.
Perfect. Good afternoon. My name is Andreas Uphoff. I work at the European Chemicals Agency, and I was asked to tell you something about REACH and biocolorants, and I'll try to do so, but don't expect too much. I'll first tell you something basic about ECHA, REACH, CLP, Classification, Labeling, Packaging Regulation, and then I'll try to apply that to biocolorants as good as I can, and maybe help you apply things to your biocolorants if necessary. Good. ECHA. ECHA is the European Chemicals Agency, established in 2007 in Helsinki, so here where we are, in the center. And it has grown quite a bit. We're about 650 employees from 28 countries, and funded from fees for registrations and by subsidy from the European Commission. We have a management board that's appointed by Council, Parliament and the Commission, which steers our actions. We have an executive director, and we have a couple of committees, which are listed on this slide, which do take the decisions about almost everything we're doing. So we prepare decisions they're taking in these committees, which are then from members from all over Europe. Good, and we have a board of appeal which checks that we're doing the right thing. Good, who are we? It's about a third Finns and then all over the place. I'm from Germany, so there are 6.5%. That gives us, oh, I can't calculate, doesn't matter, something like 20, 30. Okay, that's how much I want to talk about ECHA. Now REACH. REACH is registration, evaluation and authorization of chemicals. They have forgotten restriction. That's why it's in the last sentence there, chemicals restricted. So it's about finding out which chemicals are on the European market. That's registration. Tell us when you produce something. Seeing that people who do produce or import chemicals know what they produce and import and know that it's handled safely. If it's found that things are a bit too dangerous, then we have the chance of identifying substances as of very high concern and then subsequently potentially putting them on a list that they cannot be used unless they get authorized. And that's also checked by one of our committees. And last but not least, it's possible to restrict chemicals for specific use, like I think we're doing currently for lead shot in wetlands, and so for specific use. And Good. I'm not talking about all of these, that's much too much, but just to give you a taste. The base idea is that industry has the burden to show that chemicals are handled safely. And industry has to produce the necessary data to be able to show that, and enforcement is done in the member states. So ECHA is not going out and checking that things are done correctly. We're just checking on paper, on computers. Good. CLP, Classification, Labeling and Packaging Regulation. That's a way how to put the hazards of chemicals into boxes. So when you have a certain threshold of more than, don't know how much, 5 grams per kilogram body weight, that's a lot, then you have to classify your substance as toxic. The EU has adopted the globally harmonized system, which is a way how to label chemicals consistently all over the world to indicate specific hazards. There are some adaptations, but it doesn't really matter. Good. ECHA is maintaining an inventory of classifications for chemicals, and we have more than 100,000 chemicals now in our database. You can see them in our website, echa.europa.eu, on the bottom of the slide. And we have a body to make opinions about harmonized classifications for Europe. So we do make proposals how the classification should be, and then that is decided by the Commission in consultation. Good. Consideration for biocolorants. REACH is agnostic and CLP is also agnostic of biocolorants. There's no word about biocolorants in there, so you'll have to try to dig into there and see how it applies to biocolorants. So first, REACH is about chemicals, and they're called substances in this legislation, are biocolorant substances. The substance is defined here in Article 3.1 of REACH as something that is manufactured, roughly, including all impurities. So you make a reaction and you do some processing, and what comes out is a substance. So are biocolorant substances? Yes, they are. Yes, they are substances. Okay, now, so you are in scope of REACH. What do you have to do? I'll try to tell you in the rest of these slides what you might have to consider, but there is actually a tool on our website, and you see the link here, to help you decide what you need to do for your specific chemical. And if you do produce stuff, please go and have a look and check it out. That's better than I can ever be. Good. So first thing you have to consider, do you have to classify your substance? Okay, this is from the CLP regulation. Manufacturers, importers, and downstream users shall classify substances in accordance and so on before the tip. So yes, no condition. If you have a substance, you have to classify it. Good. Do you have to tell us about the classification you arrive at? Well, that's a different thing. So you have to tell us when your substance is hazardous in whatever way that's specified in the CLP regulation, then you should tell us. So you have to notify to ECHA that you have the substance and what classification applies. Okay, this of course causes a problem. Let's assume you have a new substance and you have no idea if your substance is dangerous or not. Should you find out or not? A chicken and egg problem. And there is some guidance. I mean, this is not in the regulation, but this is a question and answer that's also published on the website, which has been agreed with all member states. So if you don't know and you have no indication that your substance could be dangerous, then you don't have to tell us. So if you have a substance, there is no threshold in tonnage, and you know or suspect it's dangerous, then please tell us. If you have no idea, it seems you'll have to You'll have to wait for the next step when you have more obligations, and you should also find out. Good. So do you have to notify? It depends. It depends. We don't know. Do you have to register your substance? So that's the next step. If you produce or import a substance, you might have to register it with ECHA, except in some circumstances. So exempt are substances where it doesn't seem to be appropriate, and these are listed in the annex of REACH. And the annex says, one of the exemptions is substances which occur in nature. Oh, that sounds like biocolorants. If they're not chemically modified, yes. Unless they're dangerous, okay, you should know when you have to register if they're dangerous or not. And unless they're dangerous or persistent, bioaccumulative, or all of the above. And now, what is this? So, substance in nature sounds good. Not dangerous, okay, you should know. What is a substance occurring in nature? It's not maybe as easy as it might seem. It's not everything you'll find in nature is a substance occurring in nature. It's only when you get the substance by physical means, so just collecting, separation, or processed by flotation, gravitational separation, or dissolution in water, or steam distillation. If you use anything else, solvent extraction, it's not a substance occurring in nature, you're just a chemical, just a substance. And if you're not nothing special anymore for REACH. If you're a biocolorant or not doesn't unfortunately matter. So we don't know. You will have to check. Maybe it's possible that you're a substance occurring in nature, and then if it's not dangerous, you might not have to register. In most other cases, you will have to register. But good news, you don't have to register directly. If the substance is produced in below a ton per year, you have no obligation to register. And this is per legal entity producing the substance. If you produce the substance only for the purpose of research, and that includes product development, you may be able to send a PPORT notification, a process and product related development, research and development. So you have to tell ECHA that you're doing this, but you will not have to generate data. And this is an exemption. You cannot make it available commercially. And there are some conditions to this, but you might not have to register if you just do research. And you don't have to register definitely if you just produce less than a ton per year. And then it depends how much you produce, how much data you have to generate. The data is in the annexes to REACH. It's too much to list them, but of course, it goes always on top. So more difficult information requirements are at later tonnages. So the first tier is mostly PhysChem and some very basic tox, and then it goes on again. Yeah, so sorry, I'm not able to tell you, but you'll see what you have to consider. So just an example. I don't know how many are familiar with this picture. I assume you have seen it all because it's Rika's publication. So I just picked one of the examples in that publication and checked if I can find it. Now, the main colorant in this example of Tansy golden button extract is this chemical. Do I have a pointer here? Yes. So this chemical, and you can see this is an extract from our website. You can see we know about this substance already. So it is a biocolorant, but it's not a new one. It's an old substance, and it even is, as you see here, the EC number. This is a leftover from the second last chemical regulation. This is INEX listed. So it's known at least since 1980, most likely much longer. Okay, so we can find it. Oops. Yes. And, okay, you see here, this seems to be classified. So somebody has told us that there is some hazard attached to the substance. So likely you can forget not notifying. You have to notify. Most likely, I don't know if it's harmonized. No, it's you cannot say you didn't know that it might be dangerous. If you scroll down, you can see, okay, phase in. So this is old. Phase in doesn't matter anymore, actually. It was just a tiered approach on when you have to tell us about your substance, but it's over since 2018. So don't know what I put that in. We have seen notifications, as you can see here. And okay, there's some conditions. I didn't put that here. Most likely it doesn't apply to biocolorants. So Annex 3 is a list. No, Annex 3 might give you less information requirements for your registration if you have to register. If your substance does not fulfill the criteria in Annex 3, and if you see here, this is predicted to be likely to be 1A or 1B carcinogenity, miscigenity, or reprotox. So whatever you do, you'll not be released of your information requirements in this first tier. I think it doesn't apply to biocolorants because I think it's also attached to the use of substances. If it's widely used, which most likely happens to biocolorants, to colorants at all, maybe you'll have it, and it's used by everybody. Very likely it doesn't apply to colorants anyway. Okay, good. And now another complication, and then so as you see here, this thing on the left, the main compound is this luteolin, or don't read the chemical name. I think that doesn't mean it's pure. So which substance, if you produce this extract, which substance do you actually produce? Is it this main compound, or is it actually this one? We also have Tanacetum vulgare extract as a substance, which might come closer to what you're really producing. It's difficult. So it depends on how pure your substance is. And I mean, extract is not actually describing it very well. You might get different things if you extract it with some chemical or with water, I mean, with some solvent or with water, which might give you different composition. I'll stop here anyway, but it's not that easy. So if you determine that you have obligations under REACH and CLP, the next thing you have to look at is what substance do you actually produce? Is it similar to something that is near, or is it different, and how the hazards are comparing to each other? But then it actually really starts only. Yes, and if you are in this situation that you'll have to determine, and you have obligations to register, then you actually can ask ECHA. You can ask us for an opinion. You tell us what you're doing, and you send us something we call an inquiry, which is a prerequisite to registration anyway. And you can either tell us you know, what you have, or you can leave it up to us to suggest how to best identify your substance. But this is reasonably late in the process. Good, that's it. I don't know how long it took, but I think we have plenty of time for discussion.
Thank you so much, Andreas. It was really interesting, and I suppose that we might have participants on the lines who are working on this field, maybe science students or actors, and then also broad audience. So I assume that this topic is interesting for all, and maybe for broad audience it might be a new idea that biocolorants, for example, are also chemical substances that need to be.
As this whole talk is about hazard and safety of biocolorants, it's apparent that biocolorants are not inherently safer than other colorants. They might, but they might also not.
Yeah.
And that's why, I mean, it makes sense that REACH doesn't distinguish between biocolorants and other stuff, synthetic colorants, because they both might be hazardous and maybe in the same way.
Yeah.
Yes.
That's really true. So at the moment, we don't have any questions at the chat. So please, if you have anything on your mind or comments, you can write them on the chat. And Riikka Räisänen, who we have here in the studio, has raised her hand. So maybe I'll give a stage for Riikka. So Riikka and Andreas can continue the chat, and I'll monitor if you have some questions.
I also use my laptop and follow the chat afterwards.
Yeah. So thanks, Andreas, for your presentation. The last slide, not this one, but this was quite astonishing for me, since tansy is kind of one of my favorite dyeing materials, and now I see these terrible signs there, so that it's flammable, it's toxic, even this skull there, and of course also environmental toxicity and hazardous.
Self-classification by industry might depend maybe on the solvent in which it was extracted, maybe. I mean, I haven't looked at the classification and I haven't tried to make sense out of it. But it doesn't need to be due to the material extracted. It could be due to the material used for extraction, maybe, or it could be a mistake. I mean, classifications, labeling notifications are for free, and they're not always really correct.
Okay. But this really these marks. You think that it is for this is extract, because now we know also that this extract contains very many different kind of compounds. So could these signs be from this a combination of the separate compounds, or is it the extract that is?
I'm not sure.
Yeah.
I'm not sure, but as you saw here, it was predicted to be, but no, not here, but where is it? It was predicted to be potentially carcinogenic, mutagenic, or reprotox. That could explain some of these signs. I don't know if it's true, but it's predicted by, I guess, QSAR, in some computational method, I assume. Could explain it. I don't know.
Yeah, but this really shows that we also need more information about this site.
So take this information from self-classification, which we show on the website, with a bit of a grain of salt, that's the word, because these are by all actors, not necessarily the big companies, but anybody who handles chemicals, and they don't always have the expertise to notify the right thing. And because these are 5 million notifications, we don't have the resources to look at all of them and make sure they are correct. We don't have the knowledge either. I mean, for things that are not otherwise known to us by a registration, we have no data. So, yes,
yeah,
it's not sure to be correct.
Yeah,
thank you.
But thank you. This was very good example.
Yes, I'm coming here. At the moment, we don't have any more questions here in the chat, but we have a comment that the topic is very interesting. So I suppose people are taking in and learning. And Andreas will be here present. So if you have some questions afterwards, you can still write them, and Andreas can even answer them in the chat.
I can try, and if I cannot answer, there is a help desk that ECHA is providing, which also you could address your questions to. And there is lots of information on our website, which you see in the bottom line on the presentations, which you can also scroll through. And the navigator that I had on one of these slides, if you really think you have obligations, check that out, because it goes through lots of scenarios where you might have obligations and guides you through it. Good.
Yes.
Thank you.
Thank you so much. So next up in our program, we have a speaker all the way from Brazil, attending via Zoom. So we'll have presentation by Gisela de Araga Umbuzero from the University of Campinas, Brazil, and she's also a researcher in the BioColor project. And from Gisela, we'll hear a presentation about dyes in the aquatic environment. And she poses the question, should we worry about them? So, yes, now we can see well. Thank you, Gisela. Okay. Go ahead.
First of all, thank you very, very much for the special invitation in this seminar. And I'm going to tell you a story about what happened in my life. This is my life after I was introduced to the dyes. So I'm going to talk to you about dyes in the environment. Should we worry about them? I am a Brazilian. I work for University of Campinas in the Faculty of Technology, and I am a coordinator of the Laboratory of Ecotoxicology and Genotoxicity. Well, now let's begin the story. Once upon a time, in a river in São Paulo metropolitan area in Brazil, in 1998, so it was last century, I used to work for CETESB, which is the Environmental Protection Agency of São Paulo, and they have a very good monitoring program of surface water, especially the waters that are used for drinking water purpose. So almost all the rivers where water is abstracted is tested for different types of compounds, including the Ames test, which is a mutagenicity test. I can tell if the water after this test has mutagenic activity. So I would assume and think that if it's mutagenic, this water, compared to a reference site, then this is, there are some substances there that could cause this problem. Okay, so this was the river where we found mutagenicity every single month. We tested for the water and it was mutagenic. As you can see, there are some inputs of discharges in this river, and then this is this drinking water treatment plant that serves water for around, at that time, 60,000 people. Well, so this was when I was just by chance introduced to the dye. What we discovered? We discovered the source of this mutagenicity we found in the river. It was coming from a dye processing plant, and this is the river here where you can see. Let me see if I can have, okay, laser pointer. Yeah. So this is the color of the river, and this is the color of the discharge. So this material here, this water here, was very, very mutagenic, but we didn't know what was in there. So in fact, I went to the United States to develop this research in at the laboratory with Harold Freeman and at US EPA, and we found several dyes in the effluent. So this was the effluent. After extraction, you can see we find 3 different dyes. This is before the treatment of the industry, this after the treatment of the industry. Well, here is our reference site. So you can see no dyes were in our reference site, which was before all the discharges. Then 4 is just the water after the discharge of these industries. As you can see, there are several dyes in there. 5 is where the water was abstracted to be treated. Also, you see the same blue and yellow and violet, and other dyes were present. And 6, this was the drinking water that was coming from the water treatment plant. And we couldn't see any dye here, as you can see. Okay, so also we evaluated the sediment, and the sediment was, we also find, here is the reference site. We found the same dyes in the sediment where the discharge was dumped, and also at the site where the water was abstracted. We identified a compound that was, or commercial dye that we supposed that this dye was the origin of the dye in the environment. And all those samples, they were all mutagenic. The effluent was really mutagenic, the river, and the drinking water. So we said, okay, and the sediment also very mutagenic. So we said, well, maybe the dyes are involved in this mutagenicity we saw. But, so we needed to discover this, right? So our two questions were, are those dyes mutagenic that could explain this mutagenicity that we saw in the water? And also we wanted to know which dyes were there. We thought, we believed it was the ones that were in this commercial dye that they were using, but we needed to determine which dyes were there. This seems so easy, but it wasn't, because dyes are sold as commercial products, combinations of dyes, and they change the composition, because what is important is the color. So this is always very tricky. Okay, so what else we discovered? We discovered that all the three dyes were mutagenic, so they could explain the mutagenicity we saw. And also we tested the effluent in rats and observed that they could cause pre-cancer lesions in rats. So we started to be really worried, because this water was, treated water was being consumed by the population in Cajamar City. Okay, we also discovered the chemical structures. So it was the dispersed blue 373. It's an azole dye, as you can see. The CI dispersal violet 93. We're going to talk about this one later. It's also an azole dye, and the CI dispersal yellow 37. When they combine those three colors, it gives us this black color. Okay, very good. So now we are evolving. Then we started a lot of collaborations, and we were able to develop a method to quantify the dyes in the river in all those places. So what we did, we quantified the dyes, and then I summed them here. So we found, and it was very logical, as you can see, there was much more before treatment of the industry, then it was 50% only reduction, and then we found 500 nanograms per liter here at where the water was abstracted. This was the sediment concentration. Also, we found a lot of those dyes in the sludge of the drinking water. So this is good news, because it seems that the water treatment was removing the dyes that we were worried about. And we really find just a little tiny amount of the dyes in the drinking water. And we said, well, wow, how come? So why this water is also as mutagenic as the river? So we were really, intrigued with that. So we continued our research, and our question was, so what was in the drinking water that could explain, along with a little bit of the dye, the mutagenic effect? So we thought it was really a big mystery. So we went to the literature, and we learned some important things. Usually what they do when they dye the fabrics, after they dye, part of the dyes does not stay attached to the fiber. So what they do, they treat the fabric with, they do reductions of the dyes. Then the dye that is not completely attached to your clothes will become another compound. You see, this was our basic azole dye. And after reduction, it became a triazole compound. And this compound has no color anymore. So this reduction reaction will get rid of the chromophore of the substance. And then they continue to do this reduction. And what comes out of this reduction is this non-chlorinated compound, We call it non-chlorinated. I'm going to tell you why in a minute. So this non-chlorinated compound is stable and has no color, but it's a substance. And then if this wastewater, before going to the river, goes into any disinfection procedure like chlorination, it will form those compounds called in the literature like PBTA. They have PBTA 1, 2, 3, 4, depending on the azo dye that they come from. So, and they published that those two compounds were very mutagenic. So, wow, maybe this was happening in our system. And now I'm going to explain to, you know, like not scientists. I think you, in the past, when I was very young, usually when I was washing my clothes, a lot of dyes were coming off. But these days, usually this does not happen. But in fact, something is coming off from your water, from your clothes, but it's not colored anymore. So there are substances here, right? Another thing that we know about chlorination is very good to jeopardize colors, right? So this happens to everybody at home when you use chlorine during our cleaning, and we get those stains. In fact, what happened is they reacted with the dye and turn it in a non-colored compound. Very good. So then this was the mixture was solved, and we found those colorless mutagenic compounds in the drinking water that together with the tiny little amount of the dyes, we could explain the mutagenicity. Okay. Now, what actions were taken? CETESB, the protection agency, asked the facility to eliminate the dyes and related compounds from its effluents. And this was a very nice end because I believe 60,000 people were protected from the exposure of compounds that we didn't know if there was a risk or not. But based on a provincial or precautionary principle, we decided to not take that chance. So I was very happy with this happy ending. Was it the end? No, I became addicted with dyes. And then the story continues, and we found a new hotspot. Now it's, of course, also in Brazil and in Sao Paulo. This is Americana City. It's where 85 Brazilian textile production is done. So we thought, wow, let's study this place, because if the dyes are in any place, they will be there. Okay, this is how it is. This is a lot of the industries that produce textiles and also dyeing. They release in this, unfortunately, not all the industries, they send the effluent to the treatment plant. Part of them they do, but part they just release directly in this little creek here that goes directly to this Piracicaba River, which is a very important river in this area. And later on, it will be water to consumption after treatment. And also, this is the plume of the, so this is the plume of the creek, and this is also together with the plume of the water treatment plant. That is not very good in doing the job. Okay, so in this area, we found another dispersed dye, also a nasal dye, called dispersed red 1, that is also mutagenic. But then we wanted to know, wow, maybe they could cause some toxicity to the aquatic animals. We went to the literature, and there is almost no information in general on aquatic toxicity of dyes, just some, but for this dye, we didn't find anything. So we tested in our laboratory with different types of animals, as you can see, Daphnia, algae, Hydra, planaria, and also an amphipod. They represent all trophic levels, different trophic levels, and we were able to calculate what we call aquatic equality criteria. So based on all the testing we did, chronic and acute, we were able to establish a PNAC. It's the of 60 nanograms per liter, and we find in the river dispersed red 1 greater than 60 nanograms per liter. It means that we have probably the organisms are at risk. So this is what we did. We went to the river in those different sites that I showed, and we found in several sites where that were under the influence of the discharges that I showed in the previous map, that we were exceeding the value of the PNAC. So our conclusion was that yes, dyes, at least this one, is really causing, threatening the aquatic life. Okay, and we didn't stop. So this year we just did another work. It took us, I think, 3 years to do this, I believe. And what we saw, we have found dispersed violet. Remember that I told in the first slide, we found dispersed violet also in Piracicaba River here in this Americana region. But we never saw any publication if this dispersed 93 would also produce the PBTA compounds, the compounds that after reduction would make this triazole compound. So we discovered this new compound, we synthesized it in the lab, we were able to obtain grams of this compound, and also the chlorinated one. We tested them. Because of the tiny amount that we got, we couldn't do a lot of testing like with the first red one, but we tested with Daphnia, it was toxic, and also we tested with bacteria, it was mutagenic, and recently we have just submitted the paper, it has shown to be mutagenic in rats. So especially this compound in red, this was the one we tested in the rats, and it is mutagenic also in mammals, which make us very worried about. We also tested, tried to find the compound in the river. We only found this reduced form of dispersed violet 93. We looked for this one, but we learned it now that the treatment plant is not chlorinating the wastewater, so this is probably why we didn't find it. But if we, in the future, we will find, they start to chlorinate, we have already the compound to monitor. So it was found in concentrations not so much, so high, but in fact, because of the COVID, we were not able to do our sampling during the dry season where we would expect more of this compound. But even though we found the compound in Piracicaba River. So now I'm going to wrap up. Yes, I'm going to answer the question that I made to myself. Dyes in the environment, we should worry about them. Dyes and the colorless related compounds are present in the water environment, including in drinking water. They are not regulated yet. They are not regulated at all in terms of water. I never seen any standard for water, drinking water that includes those compounds. Only some aromatic amines, but not those compounds that I'm talking about. They can be present at concentrations that pose risk to the aquatic life. Textile applications, in my point of view, is of major concern because it uses high volumes of water, but paper and other applications are also relevant. And I think that we need to reduce discharges. So the less we put in the environment, the better. For me, this is the key message, because if those compounds go to the environment, they can, be concentrated in the sediment, they can stay there, they can be concentrated even in the animals. We are even able to find dispersed red one in the blood of our little shrimps. So the compound is really there, and because they usually are kind of stable, because we want them to be stable in our clothing, then this can be a problem. And of course, we would like to have the less toxic compounds, no mutagenicity. This is also another goal, but we need to think about, in order to use them, we need to have this combination of reducing exposure and reducing the maximum toxicity, which, sometimes it's not an easy task. So what is next? What about natural dyes? a new adventure now begins because of Rika and all my friends from Finland. So we are on the board of this project together. Our laboratory from University of Campinas with Wilson College of Textile, with Harold Freeman, which is my old partner, and we are together in this task. He is from North Carolina State University. So our group will be responsible for aquatic toxicity and mutagenicity of dyes, auxiliaries, and dyeing effluents. I think this is also another thing we need to deal, because of course when you're going to do all the dyeing, you are going to add different compounds. Even if they are all natural compounds, there is going to be a combination of things. And I'm really willing to try to test to see how toxic they can be, and try to calculate how much of this discharge would be safe to be, dumped in the environment. Okay, so my acknowledgement goes to mainly FAPESP, that funds the research here in Brazil, CAPES, CNPq, and Fulbright Brazil for funding my laboratory and my students and myself. And of course, CETESB, where I learned it and I worked for 22 years, and I'm very proud of being part of this team. US EPA, North Carolina University, and my university for all the support. So now I am available for questions. Thank you very, very much for your attention.
Thank you so much, Gisela, for your presentation. Very fascinating, and again, a new perspective on the effects that the dyes have on the environment. I'm still waiting for the questions in the chat. So if you have something to ask about effects that dyes can have in aquatic environments, for the environment in general, or other aspects that Gisela presented, so please leave those questions in the chat. In the meantime, I have one question that I have received through the sign-up form. So, Gisela, can you still hear me? I just want to check.
Yes,
okay.
Yes, I can.
Yes.
I mute my microphone, okay? Or I leave it on.
You can leave it on, since I'm going to ask you a question that we have received. Yes, so someone is interested about the effects of the dyes in the textile waste for the environment, and especially from the point of view of circularity. Do you have some thoughts about maybe dyeing processes where the waste waters could be circulated or handled in a way that they won't harm the environment?
Okay, can you hear me?
Yes.
Okay, thank you very much for this question. Well, it's very hard, I think, to recirculate, because dyes, they can jeopardize your textile in very low concentration. So it's very hard, I think, to do this circulation, and it would cost a lot. In my point of view, what we need is to focus on waterless dyeing process. This would be the pleasure of my life, because if you don't have water, then at least you don't have the environmental problem, right? So there are lots of applications now, like atmospheric plasma and also CO2 dyeing, where water is not involved. And I must say to you that it's not only the dyes that worry us. Why? Because even when you do some types of dyeing, you use a lot of salt in the process. And then this salt is going also to be in the water. And if you release in fresh water, this is a big issue. Conductivity goes high. And then imagine you need to take the salt out. So those processes are very expensive. So I really don't believe, of course, we can do some things, but I don't believe this would be the path. I really would love to see waterless process dye, especially with the natural dyes. I think this would be really neat.
Yeah, really interesting point of view. Would you see that these waterless technologies could become more and more popular in dyeing industries. Maybe these aren't very, or people who are listening right now might not be familiar with these technologies.
Okay, this is not exactly my field. This is from Rika and Harold, but I must say that this is in use. You know, a lot of fabrics now, like the ones I know that they make sneakers, tennis shoes, they are dyed with this this type of CO2. And so I think this technology is really working. Some dyes are not so suitable for that, so maybe we need to give a little help to nature and try to make them suitable for that. And this is going to be a REACH discussion then, because this is a natural product that has been modified to do a good thing. So I really believe that we need, that's why I think Biocolor is so special, because we have all the stakeholders involved. We need to think about less exposure and less toxicity, but all together. So everything we can do to reduce exposure of humans or animals, aquatic or even in the soil, I think this will be very nice. I think Rika can answer this thing more about, she is really involved in the CO2.
Yeah, maybe we can have Rika here to briefly discuss this matter.
Okay, about the methods. The thing is, yes, this the equipment for the non-aqueous methods for dyeing are currently quite expensive, and therefore, in most cases, because of the dyeing technology, we need water. Of course, the newer dyes are designed to be in lower liquefaction, so using less water and also lower temperatures. They are reactive in lower temperatures, and we can the new dyes also are designed for specific fibers to react well, so that there is less dye left in the dye liquor. So there are possibilities to decrease the dye in the leftover dye liquor in the effluent. But of course, as Gisela said, there are also salts present, and so also it depends on the overall industry management and how educated the workers are, how efficiently they use the machines and are aware of all these circumstances and the options in these machines that they work optimally. Okay.
Yes, thank you, Rika. There aren't any new questions in the chat at the moment. If you still have some questions, please and feel free to write those. We can also come back to them later. So it seems that we are moving forward in the program. So thank you very much, Gisela. And if you have possibility, you can still stay in the lines if someone has questions later.
For sure, thank you very much.
Thank you. So next up, I want to welcome our next speaker, Jaana Rysä, professor from University of Eastern Finland and also researcher in the Biocolor project. And she will present a topic of toxicity and human health risks of colorants. So welcome, Jaana. I will just put up your presentation here. So welcome, Jaana.
Okay. Thank you for your kind invitation. So my topic is quite wide. So it's the toxicity of the colorants. And as we know that there are, in fact, more than 10,000 colorants which are used in the textile industry alone. So I don't go really into the details, but I want to talk about the very basics of toxicity, which is kind of relevant for all kind of dyes and pigments. And to get your attention, what happens? Okay, to get your attention, I start with saying that yes, the colorants could kill you. And my example is from Victorian era, when these kind of two bright greens were hugely popular, and they were called this Scheele's green and Paris green. And why those were toxic? It was because they contained arsenic. And you don't need to be a toxicologist, but you know, if you have ever read any Agatha Christie's novel, you know that arsenic is really a convenient substance if you want to poison somebody. And at least in any English villages, they are using it quite a lot. So what comes to these wallpapers, there was this kind of dust of arsenic, and also in damp conditions, there was certain bacteria that could metabolize this arsenic compound, and there was this kind of toxic gas, and that then killed the persons. Okay, now when you are quite terrified, I can say you that this wallpaper there override is totally healthy, and nowadays not arsenic are used in the wallpapers. And also, what is my first take-home message about toxicity is it that you don't need to die. They don't necessarily, the chemicals, they don't kill you, but every adverse effect what we get from the chemicals, we say that it's a toxic effect. So, for example, skin irritation, it's a typical toxic effect, and basically not so severe one. And sometimes you don't even notice. You have, for example, elevation in liver enzymes. We can say it's an adverse effect, it's a toxic effect, but it can happen that you don't even notice that. So if we go to the textile industry and dyeing process of textiles, we have colorants, and we have synthetic colorants, we have natural colorants. For me, they are just chemical substances. There is no, I agree with the European Chemical Agency that there's not really difference between those. And we have to also remember that also many other chemicals are used in the dyeing process. So it's not only the colorants. And then after the dyeing process, the industry use chemicals. For example, formaldehyde is used to prevent the textile to wrap, and also for example, certain chemicals nowadays are used to inhibit growth of bacteria and mold. So, for example, if you buy a nice new sweater and you get allergic reaction, you can't directly blame that it must be the colorant, because it can be also these other chemicals that has been used to produce some properties that are at least economically very, very useful. So when we think the potential exposure to colorants, we have to remember that there are different groups of people. We used to think that we are consumers and we just for example, wear this weather. But there are also people who are working in the factories, in dyeing industry. And if we have, for example, a colorant which has very low or moderate toxicity, it's also possible that accidents happen. So we must know the toxicology behind these chemical substances in order that we can react in the right way if some disaster happens in the factory. And what comes to this occupational exposure, it's of course these occupational safety regulations which have the key point there. And unfortunately, in many countries where there are a lot of textile industry, the safety regulations are not at the same level as, for example, in Europe. So it really affects to that. And as Gisela already presented,
there are these wastewaters, and from those factories, we are, for example, water, we can expose also to these chemicals. So it's not only the we exposed via environment, and also I have to remind you that we are not totally innocent bystanders there, but also our actions can affect to that how much for disposal, for example, to environmental goes. So what kind of exposure we have? So probably for consumers, the skin contact is the most common way to be in contact with the colorants. And as we know, the skin is a protective barrier, and the chemicals can irritate the skin, even burn the skin. And if the chemical can pass through the skin, then we can have also systemic effects. One possibility is also ingestion. At workplaces, it's not so common to happen, but for example, if colorant is as a dust and there is not appropriate protective gear, it's possible to also ingest this colorant. And usually there are accidents, but we also have to remember that little children, you know, they want to taste everything when they are small. So that's why a special care has to be taken about those baby products, for example, that nothing dangerous is not there. And as Gisela said, contaminated water, also contaminated food can be possible routes of exposure. Then we have inhalation, and in workplaces, in factories, this is probably the most common way to exposure to the colorants. Of course, they must be as a gas, or then like small particles. They can cause local irritation in a respiratory tract, but also they can cause more persistent lung damage. And respiratory problems are quite commonly described with the persons who are working in this industry. And then we also have eye contact, and as what comes to the skin, the chemicals may burn or irritate the eye. So in order to have a toxic reaction, we have to be in contact with this chemical substance, but then kind of very large question, what makes colorant toxic, or what make chemical toxic? So basically the chemical structure is maybe the main point there. So it depends about the atoms and molecules and how they are placed in the substance. And what is really interesting that we know that if the chemicals have similar structures, we can assume quite many times that yes, they have similar toxicological properties. But on the other hand, we know that there can be only slight differences in the chemical structure, and there can be huge differences in toxicity. So these things are not too simple. And probably the main issue in whole toxicology is the dose. So how much of substance we are exposed to, that also tells or kind of describes what kind of effect we have, or how severe this effect is. And our body, it also have ability to change these chemicals into the less harmful forms, even eliminate them in the body, but sometimes these processes does not work, or in many times they don't even exist. But anyhow, what makes the colorant or chemical toxic, it means that it has the ability to change some function or structure or maintenance function of the cell in a way that it's harmful for us. And here it's quite a busy figure about what can happen, but what I want to demonstrate with this is that if we have for example impaired, what happened? So impaired ATP synthesis or calcium regulation, what we see as a functional level is that the cell function is not working normally. So why toxicity has to be studied in detail is the reason for that is that there are so many mechanisms of toxicity. And at this moment, we don't have one single test we could really see every possible toxic reactions, but we have different tests to different class of toxicity, and that's why many different experiments are needed to really fully see that whether chemicals are harmful for us or not. Okay, I'll try to go to the next slide. Okay, now, so if we now go from the chemicals to specifically to colorants, so what are the main effects that's been described? So there are local effects like skin irritation and skin sensitization, and then we have systemic effects, mutagenicity, carcinogenicity, neurotoxicity, and they can even disturb the hormonal system. And if we talk about the skin sensitization, which is maybe one of the most common adverse effects related to colorants, this skin sensitization means that person becomes allergic to a colorant. And here, if you take a closer look at this table, you see that there are different kind of textile materials, and then there are different dye classes, and then there are column which tells how often or how potent allergens these substances are. And what is really interesting is that we can have the same dye class, but if we have a different substrate, the allergic reactions are rare, or they can even be frequent. And as you see there, that some allergens are described only as occupational allergens. So it tells that you really must expose quite a lot to this substance. So what is rare and what is frequent? Well, it's quite difficult to say, but I found one study done in Denmark when the estimation was that maybe 2 persons out of 1,000 who have eczema, which means kind of a allergic skin reaction, is caused by colorants. And I think this article didn't say, but I assume that the main cause is hair dyes, because the hair dyes especially are quite well known to contain one very potent allergen called PPD. But so these allergic reactions, these are not related to textiles, so they are quite rare. And also I would assume that it's quite difficult to also, if you go to the local healthcare center, to convince your doctor there that this must be the colorant which causes this allergic. So I also could imagine that there are some problems to how to diagnose these. Gisela already mentioned the mutagenic effects, and why we are really interested in these is because mutagenicity is always a serious harm. So what it means that the substance causes genetic damage, and they can change how the cell is functioning, but also they can be passed to the new cells and they are formed already as a damage form. And what makes this really why also like European Chemical Agency takes these mutagenic and substances really seriously is the fact that they can also result in cancer. And one thing is that most chemicals that cause cancer, they also cause mutations, but not all chemicals that cause mutation cause cancer. And also we know chemicals that don't cause mutations, but they cause cancer. So this is not too simple. And here there is this figure about bacteria plates, and this is kind of nice and way to study about the mutagenicity of chemicals. So we are growing bacteria, and if the bacteria is mutated, there are more colonies there, as you can see as the plate there on the right side. And then we go back to the basics. So I know that our project aims to develop a nice natural colorant, but my one take-home message is that if you have a natural substance, it doesn't mean that it's harmless. And I want to convince you, and at the left side I have this figure which tells how toxic this kind of synthetic and natural chemicals are, and the most toxic chemical we know is botulinum toxin, and as we know, not any evil chemist has made that, but it's perfectly from the nature. And then if we go to this, that what is natural and what is synthetic, it's not so easy to say. If we eat oranges, we know that they have plenty of vitamin C, and it's really healthy and it's natural. But then if we, for example, use glucose and we made vitamin C from the glucose, then we have synthetic vitamin C. Basically, the chemical structure is the same, but the other one is natural and the other one is synthetic. And if we go to the process of how to make alcohol, we can have even more complexity there. So we know that alcohol can be formed naturally in fermenting fruit. We know that if we have ethane and steam, we can make alcohol, but we have also process when we control this kind of alcohol fermentation, which is kind of borderline exception here. So my take-home message, natural is not synonym to harmless, and synthetic is not synonymous to toxic. It can be all the possible combination of these words. So why natural colorant? So at the right side is a nice figure. So you maybe see this kind of willow bark, but I see these chemical structures which are there over the right side. So it's well known that many toxic and hazardous chemicals and very harsh conditions are needed to produce these synthetic dyes. And these natural dyes could offer a sustainable alternative, but we must be careful, and detailed toxicological studies of these new dyes are needed in order that we can establish that they are really safe both to the humans and to the environment. So there are a lot of possibilities and better options, but we don't know it before we study that. And in this study, in this BioColor project, we are studying toxicology as a cooperation of 3 different universities. There is a State University of North Carolina in US, which is taking care of the mutagenicity studies. As Gisela presented, University Campinas in Brazil is in charge of environmental toxicity studies, and University of Eastern Finland takes care of the human health, and we are carrying experiments on human cells. And here is our team in Kuopio. And I want to thank you. And if you have any questions, I try to answer them. Thank you.
Thank you so much, Jaana, for this interesting presentation and opening the conversation towards the human health. Green here. We have at least one question concerning your presentation. It might be that someone else can also elaborate on this question, but there's a question about the black color and its effects on body. Do you have anything to say about that? If like certain colors or colorants are more?
We can also make many options for black colors, but what I know, what is really concern, it's black henna. So this natural product is this kind of red henna. It's quite harmless and quite not allergic at all. But in this black henna, there is also this chemical. Abbreviation is PPD. I don't remember what is the long name, but it's really highly allergic. And that's why there are lots of instructions that please do not go and don't take any black henna tattoo, because you know it's there in contact with the skin for a long time. And because almost all, not all, but I mean very huge percents of hair dyes, synthetic hair dyes are also containing this PPD. So when you have first this contact with your tattoo to this PPD, and then you go to the hairdresser and take a hair dry, you will have a huge allergic reaction. And this PPD, maybe our expert from European Chemical Agency could tell that it's kind of restricted in EU. I would if I could remember correctly, in hair dyes it is, and there is this certain percentages of what is accepted, but these henna tattoos, they are not something we like to have. But what comes to the other possible black colors, maybe Riikka knows better.
Yes, I can give space for Riikka.
Yes, I would say that there are lots of different dye structures for black color, and these different dye structures, of course, since they are different chemicals, they may also have different characteristics in terms of toxicology, and therefore it is quite difficult to say if we don't know what type of dye is it particularly that is used in some application, and for different fibers different types of dyes are used. But for dark colors anyway, the option to get more irritation, for example, is due to larger amount of dye in the textile already, because if you have a light color textile, there is less dye in it, so it is obvious that the possibility to get irritation is lower.
Thank you, Riikka. We don't have any more questions at this point. Okay, yeah. And again, feel free to still add the questions, but I give room for Riikka again.
Okay. I was thinking about, you was talking about this that the chemical will interact with the processes in the body, in our body, and this makes the harmless or toxicity. What about, I was thinking of dyes, some of the synthetic dyes, they are very stable, even they might be even inert, that they don't cope with anything. Would this kind of substance be non-toxic if it's inert, that it's not coping with anything around it?
Well, basically it excludes, of course, certain kind of reactions, but it's possible that this kind of chemicals could also accumulate. And we don't know that, for example, if they disturb some processes, for example, the structure is so similar to some substance which is naturally in our body, so it kind of fools our body to use something else. But of course, if we have a less reactive dye, for sure, it's not. I mean, it's a good start. For sure, it's you could assume that it's not that toxic. But we know that, for example, in liver, we have these metabolizing enzymes, and they have an ability to move, to transform the substances also to more reactive form. So it's also a possibility in that way. So basically, these liver enzymes are doing good. So if we have toxic substances, they are making them less harmless, but it's also possible that they are making more reactive forms, and then we face some toxicity problems.
We have a new question here. It might be to some other of you, but I will just read it here. So, is there a battery of tests that could be recommended for clothing, taking account the heat, stress, wear and tear of washing, interaction with chemical treatments and washing agents, and interaction with compounds of skin and sweat, packaging, waste processes? This is a long question, and it still continues. So, how useful is QSAR in designing modified bioproducts? It helps. You can also see it here.
Okay. So yes, it was a long question.
Maybe long and specific.
Yeah. So, well, to be honest, I don't know the answer, but what I can tell here that, for example, when we think about colorants, it's also what is where we use them. So, for example, food packaging, clothing, baby bottles, baby clothes. We have different ideas how safe they have to be. And for example, some colorants which are used in paints, they are absolutely forbidden to use, for example, any playground, because it's possible that they are flaking and then the children are eating them. But for example, in the factory, it's quite okay to paint the floor, because we don't really accept that these men working there are licking the floor. So it's also how we use the chemicals can make the difference. So we can have more toxic chemicals if we use them wisely, and if the environment where we use it is appropriate, and especially the persons who are using the chemicals know what's going on there. But for this kind of clothes, I don't really working in the clothing industry, but I would say that, and maybe there are not these kind of patterns, batteries of tests, but of course when we have chemicals, then REACH gives the regulations. It depends about how much this chemical is produced, and then there is certain battery what you have to test. But this is really an interesting question. And for example, these skin sensitization reactions, there was some articles that they are more common in humid conditions. So when I said that in Denmark they are quite rare, they can be much more common in humid conditions. And why is that? It's not totally known. So it can be that they are wearing more synthetic clothes, but it's also possible that because it's humid, maybe more dye or other chemical is going to in contact with the skin. So, but we don't really know that. And what comes to this QSAR, so I don't know. I think that now our chemists from ECHA can answer this question.
I cannot answer that question. Of course, QSAR can. Sorry.
So yeah, let's welcome Andreas back on the stage.
Not be an expert in QSAR, but of course QSAR can be used to try to predict properties of chemicals based on the structure. And that is not only applicable to synthetic chemicals, but also to biocolorants. So yes, you can try to apply QSARs to biocolorants. Of course, these methods need to be developed and verified. So I'm not sure if this exists already for biocolorants, for the chemical classes that are of your concern, of interest. It may be that they're not readily available, but in principle they can be used, yes. I don't know how often you do modifications to things you find in nature. Of course, it's not, at least for me, I don't know how you would predict what kind of chemicals you get out of your plants or fungi. So there it wouldn't help you. As soon as you know what you get and somebody has verified a QSAR method for this category of chemicals, why not use it? Yes.
And I think that the benefit of QSAR is also that because there is, I can say, enormous amount of chemicals if we think that we should test them all, I mean, in every detail. So QSAR could help us to identify those chemicals which we already know that could be toxic. And that would mean that we have to do less testing because it's really, you need a lot of money, you need a lot of time, it's not good for the company, and it's not good for animals, it's not good for anything. So I think that it also helps in that we can, if we can already say that this looks so suspicious, this is so looking similar that these toxic structures in other chemicals, so we just exclude them, and then we have a certain chemicals which then go through the test battery.
May I make a comment on the QSAR?
Yes, I was coming to give you a moment of speech, so please.
Okay, well, I have been trying to use QSAR for mutagenicity and for aquatic toxicity quite a lot. What I recognize today is that QSARs are based on previous knowledge of information on toxicity and mutagenicity. And dyes are a very tricky category. Why? Because a lot of the toxicity tests, especially mutagenicity, the data is really old. So at that time, like in the 80s, they didn't have the ability to test pure substance. So there was a lot of data in the literature that they don't even say what was the purity of compound. So imagine you were trying to match toxicity with the chemical structure of the substance. But if you have data that is kind of misleading, then you have several problems. And if they are too general, if you have one single, like a NO2, like an alert in the substance, then it will come up as positive. So in my experience, we still need data for dyes. It doesn't matter if they are natural or not. I'm talking about anthraquinones, I'm talking about other dyes that they are in nature, but they have been tested not so poor. So it's very hard to use QSAR these days, specifically for dyes. But on the other hand, this is the way to go. So I really think we should go forward. I have one paper published on QSAR for ecotoxicity. I can share if you guys want here in the chat. It's an open access publication. Then I think this can give an idea of where we are now in this QSAR thing. I think it's very useful, but we need to still have some data to put it on in the database and have better predictions. This is my opinion.
Thank you. Yes. Actually, you also posted a comment in the chat earlier that you had some question for Andreas. It seems that we are kind of moving from Jana's presentation to general discussion. So I want to thank Jana at this point. So we have still time for general questions we have already some of those, and you are still very welcome to leave new comments and questions. But Gisela, if you had some question, you can pose it now.
Well, thank you very much, Andreas, for your talk. It was really very nice to have you here in my room in Brazil. And I have a question that I didn't fully understand. The compounds that are below 1 ton. So what are the requirements? If I have a compound that I'm testing now, a new dye, how should I proceed? Do I need to register, or the registration means that I will use as a commercial product? This part is a little bit, I understand better when it's more than 1 ton, what I need to do. I know the batteries, but it's not clear to me what I do when I have something in the market or not that is going to be below 1 ton. Can you clarify it for me?
So you personally are not in the EU at all, I think, so you wouldn't have to do anything. If somebody buys your substance and imports it into the EU, then the person importing, if they import more than 1 ton per year, will have to register. So if this substance is only above 1 ton per year, per manufacturer or importer has to be registered. The classification and labeling and the notification for the inventory is independent of that. That has to be done also at lower tonnage if the substance is hazardous, known to be hazardous. If you use your substance, almost regardless of tonnage, only for research purposes, then you can avoid registration by sending us a P-port notification, but also only if you are acting within the EU. So whatever you do is not touched by anything ECHA has authority over. And if you cooperate with somebody in Finland, for instance, and send them more than 1 ton per year, yes, then you have to, then the receiving company has to register. So you don't have to do anything. It would be nice if you provide the data known to you about the substance. But,
thank you. Thank you very much. I still have a comment. In fact, what is happening in Brazil and other countries, we are kind of following REACH, not in the registration, but for sure in the GHS system. So we are adopting it. So what you were saying to me is that the classification is something I need to do, or I can do whatever. So registration is a different thing. Registration is something legal if I'm going to send or import or send to Europe. But in terms of GHS, then we are going to use the same classification. And then the tests are quite the same, or part of the tests that we do usually are based on the ones that are required. So is my understanding correct? So classification is completely separate from registration. Is that right?
Separate. The classification is necessary before registration. It's always necessary for registration. So if you register something, you also have to send us a classification with that. If you don't have to register, you always have to classify. You may have to tell us if you're acting within Europe.
Okay, thank you. Now it's really clear.
GHS, I think, is globally harmonized, but there's slight adaptations, I guess, per region. I'm not certain exactly. It's mostly the same, but not in all details, if I get that right, but I'm not an expert there.
Okay, thank you very much.
Good, thank you.
Thank you. We have still a few questions. So I'm actually trying to combine a couple of questions that I assume might have been posed by someone who dyes with botanical dyes or natural dyes in their crafts. So there have been a couple of questions about botanical inks used on paintings or applied on paper, and also yarn dyed with natural dyes. So how one can assure that those are safe And for example, if some mushrooms that are toxic are used for the dyeing, how one can assure that the yarn is safe to use. I suppose this might be Riikka Räisänen's or
I can answer.
okay, thank you.
Yes, I have studied some textile woolen yarns that have been dyed with natural dyes. Of course, if we think about plants or mushrooms that are toxic, so I think it was very good, was it in Jaana's presentation, that we have to think about, do we eat them or are they on our skin? Because like in mushrooms, there are these toxins. For example, this some of the Cortinarius species are very toxic when we eat them, but some are used for. They are also good for dyeing. Not the same species, but different species. So it's always also very good to know what species you are collecting. But anyway, in dyeing, there are compounds extracted in water, and then from water they go into the fiber. And we have tested these woolen yarns and extracted compounds from the yarns and tested the toxicity in some simple tests. But those showed that the levels of the compounds in the extracts are so low that they don't cause harm for the user. So and of course it's always also on the skin of the person. So more probable thing is that the irritation might come from the fiber or from other substances that are present in the fiber than actually from the natural dyes that are in the fiber. Do you, Jaana, want to say something?
Yeah, so a couple of points, and first is of course that you probably don't dye like every day and many hours per day, so it's kind of occasional, so the exposure is not constant for those. And also when you do the exact dyeing process, you probably wear gloves, and maybe the face mask nowadays is also a good option, so then you can diminish how much you expose. And as Riikka said, then in the final product, the concentration or the amount of this substance is really low. And also that, Riikka can correct me, but usually there are historical colorants what are used. I mean that people have traditionally used them like 100 years ago, maybe 50 years ago. So there are lots of knowledge, and probably if some dyeing experiment has caused a really severe skin irritation, it has not come highly popular. So I would say that if you have normal cautious, you don't drink any substances there, and if there are some gases, you don't inhale them a lot. So I would say that it's quite secure. But if we think the colorants, as I said in my presentation, we also have to think about those who are working in the textile industry and also in other countries than Finland, and then the working conditions are already totally different. And that's when the dye, which is not so toxic, if it's used correctly, can cause several severe health hazards.
Yeah, and then there was a question concerning these botanical inks, and yeah, for printing cards and drawings. I would say that if you extract the ink or the dyes
colorants from these plants and then use that for drawing. I would say that it is safe, but I wouldn't eat it. So everything that is outside our body we would consider safe, but if we do the extractor dyes, I wouldn't eat it before we have more knowledge of biocolorants toxicity for digestion. Of course, then you can find from these E-codes for natural dyes. So there are natural dyes used in food quite a lot actually, and of course those dyes are then safe also to be eaten. So and from vegetables that we eat and if we extract the color for them, it would be even of course safe to eat them. And well, there's some other.
Yeah, I could comment this. So still adding to this conversation, we had a comment earlier in the chat about the luteolin that was raised in Andreas Uphoff's presentation, that luteolin is flavonoid, and it's also found, for example, in nettle seeds that are considered as a superfood and very healthy. So this comes also to the question whether we are eating some some colorant or compound or exposing it to the skin, for example. We've had a very interesting question concerning the waterless dyeing methods. I suppose many of you there might find this interesting. So the question is, are there any research about waterless dyeing methods on natural fibers, on cotton, wool or silk? The person who poses the question has heard that these CO2 dyeing methods have been used on polyester, but what about natural dyes? Natural fibers?
I remember that I have written some research on using CO2 for natural fibers, but because of this technique, the way it functions, it works the best for thermoplastic fibers. So when in the cases when this fiber structure softens and melts in these conditions, and the dyes go as small particles inside the fiber, and this is the reason why most applications are on polyester, for example, but other thermoplastic fibers would work as well.
Okay, thank you, Riikka. It seems that we don't have any new questions in the chat at this point, and actually we are right in the schedule. The webinar is coming to its end. If you still have some very important, interesting question, feel free to put it in the chat. But at this point, I will share the last slide of our webinar. So about how you can keep up with the project and follow us. Okay, so here you should see all the information about Biocolor Project's social media channels, also our website. Perhaps many of you are already familiar with these, but by following us through these channels, you'll receive information about proceedings in the project and also about the future events and workshops we are organizing. And if someone is wondering if this webinar will be available later, so yes, it will be added on our YouTube channel, most likely in the end of this week. So you can always come back to the information shared in this webinar, and if you have some contacts, for example, who might be interested on these topics, you can tip that the webinar is available later. It seems that it's time to wrap up, so thank you so much for all of you for active participation, and I hope you got lots of new interesting information. And maybe the conclusion of this webinar is that the critical research and testing is very crucial also in the case of natural dyes, since we can't be sure that those would be safer than any other dyes. But thank you so much for joining. Bye.